hands. So good morning, everyone. Welcome to the Department of Surgery Grand Rounds. It's a little bit of a rainy day, but hopefully everyone has had a nice start to their morning. We are very fortunate to have our own Dr. Meghan Flanagan with us this morning to present. She is certainly not a stranger to most of us here in the department. Dr. Flanagan started her medical career at Albert Einstein and then actually came here to the University of Washington for her residency in general surgery. Also completed research time and a Master in Public Health during that time. She then went on to Memorial Sloan Kettering for a breast surgical oncology fellowship, and we were fortunate enough to have her return to join our faculty in 2018. As I would say is very typical for her, she has been highly productive and effective since her return here to the University of Washington. She has her own grant as of this year, 2020. She has been an author on over 7 peer-reviewed articles just in the last 12 months, which is incredibly impressive. And also has been very active in growing the breast and surgical oncology breast program at Northwest Hospital through the Hutch. So Dr. Flanagan, thank you so much for being here this morning. We'd love to hear from you.
Thanks so much for the very kind introduction. Sounds a lot more impressive than I feel. So I think for better or for worse, I'm going to be giving a lot of background today. Getting translational research up and running in the area of COVID and also having a baby has been slow, but I'm really excited to share with you the work that I'm planning on doing at least today and some of the background for it. So the title of the talk today is The Role of HSD3B1 in Postmenopausal Breast Cancer, and we'll learn what that is as we go along. So I'm sure a lot of people know this, but breast cancer is the most commonly diagnosed malignancy in the United States among all comers. In 2020, it's estimated that there'll be about 276,000 breast cancers diagnosed. And among men and women, breast cancer has the 3rd highest cancer-related mortality, and among women, it's secondary only to lung cancer. In terms of thinking about who breast cancer mostly affects, this graph just shows the new cases, % of new cases on the y-axis and age of diagnosis on the x. And you can see here that the vast majority of patients who are diagnosed with breast cancer are in the postmenopausal age range. So about 70% of women are over the age of 55, with the median age of diagnosis at 62. And the reason I share this is that my research is specifically focused on postmenopausal women with breast cancer. So that's the majority of women who do get breast cancer. When we think about how to classify breast cancer, there are the histologic subtypes that we all learned about in medical school, so invasive ductal and lobular cancers. But the most important part of thinking about classification of breast cancer is actually the molecular subtype or the biology of the disease. HR here means hormone receptor. Some breast cancers, the majority of breast cancers actually, will either express the estrogen receptor, the progesterone receptor, or both, and that's HR positive breast cancers. And then some breast cancers also can be overexpressors of HER2, and that's HER2 positive versus non-overexpressors or HER2 negative. And the significance of this is that the vast majority of patients who have breast cancer have hormone receptor positive breast cancer. And this has implications for treatment because for those patients, we know that estrogen is actually stimulating the cancer cells to grow. And so if we can block the body's production of estrogen or the interaction of estrogen with its downstream receptors, then we can hopefully prevent recurrence or progression of disease. So this is actually a simplified version of androgen and estrogen biosynthesis, but we really need to have a pretty good understanding of this to be able to understand my research and how it works. So I'm just going to take you through synthesis of estrogen here. So premenopausal women who have ovarian function intact, the ovary is the primary producer of something called estrone, which is converted to estrogen, estradiol, which is E2 here. Estradiol then can go on to interact with the estrogen receptor. This acts as a transcription factor to transcribe genes that have to do with tumor proliferation, cell adhesion, things like that, that can actually lead to tumorigenesis. In postmenopausal women, when ovarian function declines as we age through menopause, there is still estrogen production in the body, but it comes through a different route. So this is primarily through adrenal production of androgen precursors. So DHEA, testosterone, and 4-androstenedione go through a series of reactions to ultimately be converted to estrone and estradiol, and then go on to have the same interaction with the estrogen receptor. So for women who have hormone receptor positive breast cancers, estrogens play a significant role in the development and progression of the breast cancer itself. This is primarily through the interactions with estrogen receptor alpha. And the significance of this is that we have targeted treatments that can dramatically improve survival and progression for these patients. There are different ways that we can sort of decrease estrogen in the body. That's decreasing extragonadal estrogen synthesis through a class of drugs called aromatase inhibitors, and I'll show you how those work in just a second. We can also actually block the interaction of estrogen with the estrogen receptor, and that's a class of drugs called SERMs, or selective estrogen receptor modifiers, or a drug called tamoxifen or raloxifene. And then there's a newer class of drugs called SERDs, or selective estrogen receptor degraders, which actually bind to the estrogen receptor and induce its degradation. So here's that same slide again, just slightly more complicated. Premenopausal women are in the yellow at the bottom here. The only drug treatments that we have for them are the ones that actually directly interfere with estrogen binding to the estrogen receptor. So tamoxifen and a drug called fulvestrant, which is a SERD, are used in premenopausal patients. Fulvestrant is actually an injectable medication, so it's not really prime time for adjuvant treatment for 5 years, as it needs to be injected every 2 to 3 weeks. So this is really only approved in the metastatic setting. But when we look at postmenopausal women, they have, we think about a class of drugs called aromatase inhibitors. So aromatase is a really important enzyme in the production of estrogen for postmenopausal women. It's the enzyme that catalyzes the rate-limiting step, converting testosterone and androstenedione to estrogen. And this class of drugs, aromatase inhibitors, essentially interferes with the binding of these adrenal androgen precursors here to aromatase, so the estrogen cannot be produced. And if you look at estrogen levels in women who are on aromatase inhibitors, they're almost undetectable. This is the class of drugs that most patients, almost all patients who have postmenopausal breast cancer get placed on, both in the adjuvant setting, meaning after surgery, also in the metastatic setting. So really important class of drugs. Unfortunately, about 25 to 30% of women are resistant to endocrine therapy. You can either have intrinsic resistance, which means that there's something about you that doesn't allow you to respond well to endocrine therapy before having even seen any of the drugs. And you can also acquire endocrine resistance, which means that under pressure from receiving one of these treatments, your tumor sort of subverts the sensitivity and becomes resistant to the drug. There are various mechanisms by which someone can be resistant. These represent sort of acquired resistance. Ligand-independent estrogen receptor activation basically just means that somehow the estrogen receptor is just permanently turned on. It doesn't need estrogen to bind to it to be on. This is mostly through something called an ESR1 mutation, or you can have growth factors that will phosphorylate the estrogen receptor and lead to activation. Some breast cancers lose estrogen receptor expression, so no longer become sensitive to hormone endocrine therapy. And there's also a variety of other cell cycle and cell growth pathways that can be activated in these patients. My research actually has to do with intrinsic resistance to endocrine therapy, and we'll talk about that in a little bit. So I start this slide because despite the fact that the majority of patients who have breast cancer are hormone receptor positive, and the majority of patients that are hormone receptor positive receive aromatase inhibitor therapy, we actually don't really understand, and there are a few biomarkers that can actually predict a woman's response to endocrine therapy and or our ability to predict who will become resistant or is already resistant to endocrine therapy. So this reflects a lot of women who get this kind of treatment. And so there's a real need to study alternative agents or novel targets so that we can treat these endocrine resistant tumors or identify patients up front who we think are going to be resistant. So I'm going to move into prostate cancer here. I'm definitely not an expert in prostate cancer, but this serves the basis for my research. Prostate cancer parallels breast cancer in a lot of ways in that it's a hormonally driven cancer. Almost all prostate cancers express something called the androgen receptor. So breast cancers express the estrogen receptor and prostate cancers express the androgen receptor. Growth of prostate cancer cells is really dependent upon stimulation of the androgen receptor binding by DHT or testosterone. And similar to women who have aromatase inhibitor therapy or SERMs or SERDs, men undergo something called androgen deprivation therapy. So essentially the goal is to decrease the amount of androgen available in the body. So this can either be done by surgical castration, removal of the testicles, or by chemical castration, which basically shuts down testosterone production. Unfortunately, most men actually ultimately develop castration-resistant prostate cancer, meaning that they are also no longer sensitive to the androgen deprivation therapy. So same little diagram, but this time for men instead of women. So the estrogen part of it is not there, obviously. In men, testicular function does decrease over time, so there is a slow decline in testosterone production, but generally over the lifespan, most of the testosterone production does come from the testicle. And then they also have this adrenal precursor pathway where DHEA is produced by the adrenal gland, metabolized to androstenedione to testosterone, and through an irreversible reaction with 5-alpha reductase to DHT. Dihydrotestosterone is an extremely potent androgen receptor agonist, meaning it binds and much like estrogen to the estrogen receptor will work to promote tumor progression and tumorigenesis. So this is the gene that we're going to be talking more about today, this is the enzyme that comes from the gene we're going to be talking about more today. In the adrenal androgen precursor pathway, so DHEA to androstenedione and testosterone, this is the active enzyme. It's called 3-beta HSD, and this in men is the rate limiting step in producing testosterone. So I'm going to take you back a little bit to some Mendelian genetics. The gene that we're going to be talking about is HSD3B1, and the gene product is 3-beta HSD1, which I just showed you. So when we're all born, we have 2 copies of each gene. And in this HSD3-beta1, the homozygous wild type is the base pair at this specific position in the gene is an AA. You can either have a germline. Germline means all cells in your body can be have a 1 base pair copy change from an A to a C, and that's called a single nucleotide polymorphism, or a SNP. You also could be born with having the homozygous variant, so having 2 copies of the C in all of your cells in your body. The significance of this, the N here is the amino acid that's produced at this base pair, and the amino acid has to do with the enzyme itself. So here there's an asparagine. When the A switches to a C, it becomes a threonine. And the significance of all this is that you can be born with either the homozygous wild type, heterozygous, or homozygous variant, or in your cancer, if you have a cancer, in the cancer cells, you can also have a mutation, and you can be germline, or all cells in your body wild type, have a mutation in your cancer to become heterozygous, or you can have a loss of heterozygosity to become the homozygous variant. And let me show you the significance of that single base pair change. So this is a really nice illustration of how exactly the single base pair change works. In patients who have the mutant or variant copy, so the A turns to a C, this enzyme 3-beta HSD becomes resistant to degradation in the cell. So over time, this enzyme continues to build up in higher and higher concentrations, and you can see that these reactions are one way. So DHEA is driven with increasing concentrations of this enzyme to produce more androstenedione and testosterone. And you can see this very nicely. These are some in vitro lab experiments. Here on the y-axis is the percent of the various androgens in the cell in terms of concentration and percent. And on the x-axis is the time of the experiment, 0 to 48 hours. So let's first talk about people who have the wild type. And this is just looking at either germline or somatic copies. So the wild type means that their enzyme is not is not resistant to degradation and can be degraded in the cell. And so we'd expect to sort of just progress evenly and slowly through DHE as metabolized to 4-androstenedione. So DHEA slowly goes down, 4-androstenedione slowly increases over time, and then through another set of reactions becomes ultimately DHT, and you can see here slowly DHT increases in the cell. Very different look than the person who has the variant. So this is 2 copies of the variant allele. This means that this enzyme is resistant to degradation, and the pathway gets sort of driven through increased concentrations of this enzyme in the cell. DHEA over time dramatically decreases in the cell as it's metabolized to 4-androstenedione, which you can see dramatically increases. And then as it goes through the series of reactions, irreversible reaction to DHT, DHT dramatically increases in the cell. And as I had previously mentioned, DHT is a very potent androgen receptor agonist. And so in men who have prostate cancer, this is very significant in terms of their potential for progressing past androgen deprivation therapy, as DHT sort of is an extragonadal, DHT has extragonadal synthesis to promote tumor proliferation in these patients. So always we want to confirm that what we find in the lab is actually clinically significant. So the same group who did all of the lab work in developing and finding this SNP did a multi-cohort study with 3 separate cohorts of prostate cancer patients. The primary cohort and the post-prostatectomy validation cohort were men who had a prostatectomy and then had evidence of either biochemical or clinical failure on ADT or chemical castration. They also looked at a metastatic validation cohort, was 188 patients who had metastatic castration-resistant prostate cancer and had blood samples available for research. In order to sort of establish what the allele frequency is of that C copy, they germline sequenced all of these patients and found that the allele frequency of the variant or the C copy was about 29%. So that's a pretty significantly high number of people who already were established to have this variant in their germline. They found 50% of patients were wild type, 43% heterozygous, and 7% had the variant status. In larger databases, just population level, this allele frequency ranges from about 10 to 35%. So this is right in line with that. And it does vary by race. So their hypothesis was that men who have the homozygous variant allele, so meaning 2 copies of the C allele, would have all this extragonadal synthesis of DHT, which would promote tumor growth and progression from past androgen deprivation therapy to castration-resistant prostate cancer. The wild type allele they thought would have the best outcomes, and the heterozygous patients they thought would have intermediate outcomes. In each of the different cohorts, the demographic and treatment characteristics really didn't differ according to genotype, with the exception that in the post-prostatectomy validation cohort, more of the men who had the homozygous variant allele had N1 disease as opposed to N0, and they accounted for this in their multivariable analysis. So this is a Kaplan-Meier curve. The percent of patients who had progression-free survival is on the y-axis. The years of follow-up is on the x-axis. For those of you who are not familiar with Kaplan-Meier curves, 100% progression-free survival happens at year 0. So this is just after someone has surgery. And then over time, more and more men would develop castration-resistant prostate cancer, and the numbers of progression-free survival decline. You can see here, this is pretty dramatic, but patients who had the homozygous wild type had the best progression-free survival here in green. Heterozygous had intermediate progression-free survival, and those who had the homozygous variant, so 2 copies of the variant allele, had the worst survival. And this was all statistically significant. And just sort of a different way to think about this, this is median survival or 50th percentile of survival. And you can see here the homozygous wild type had about 6.5-year median survival, heterozygous 4.5-year survival, median survival, and the homozygous variant 2.5-year median survival. And I just want to point out that in cancer research, we're often happy with having improved survival on the order of months. And this is really significant because this is improved survival in the order of years. When they looked at progression-free survival in the other two cohorts, or the validation cohorts, they found more or less the same type of results, where patients who had 2 copies of the homozygous variant in red here and in red here really had the worst outcomes. In these particular cohorts, there was not statistically significant differences between the heterozygous and the homozygous wild type. But I will mention also that this is only germline sequencing, so this study didn't specifically look at any mutational changes in the actual prostate cancers themselves. Again, just to drive home the point, this is looking at overall survival in the primary cohort, and you can see dramatic differences in overall survival here. At 10 years, 55% of patients who had the homozygous wild type were still alive, 35% of people with the heterozygous 1 copy of the mutation were alive at 10 years, and none of the patients with the homozygous variant were alive at 10 years. So their conclusions were that they were able to validate their hypothesis that the homozygous variant had significantly worse prognosis in progression-free, distant metastasis-free, and overall survival. And the clinical implications for this is that we may be able to either germline sequence patients up front, or once they've had surgery and we can sequence their prostate cancers, we can identify some patients up front who may be more resistant to androgen deprivation therapy and figure out ways that we can escalate their therapy earlier. So potentially give them some cytotoxic chemotherapy or add anti-androgen therapy onto their treatment regimen. And this is that same diagram again, but showing some different drug targets available in prostate cancer. So abiraterone is a 17 beta HSD antagonist, and enzalutamide and bicalutamide are androgen receptor antagonists, and both of these have been shown to improve survival in patients who have androgen receptor positive castration-resistant prostate cancer, specifically people who are metastatic. So you can see that there's a significant clinical implications for having this mutation in terms of survival and prognosis, but also potentially some ways that we could change these patients' therapy up front. So that's the background and how we sort of were kind of what I'm basing my hypotheses and research on is based on what we see in prostate cancer. So what do we know about this gene in breast cancer? And I'll just start by saying that we really know very little. There's almost no data in the literature looking at this. These are 2 studies. One was done in China and one is done in Japan. And they were looking at actually just expression of these genes in breast cancer cells. And they looked at hormone receptor positive cancers and negative cancers. And they just looked at immunohistochemical staining of the cancer cells and whether they expressed the gene or not. So it has nothing to do with the mutation. It's just, did patients express HSD3B1 and what was the significance of that in terms of recurrence-free survival? And these 2 studies are all that exists, and they had exact opposite findings. So the graph on the left is another Kaplan-Meier curve with recurrence-free survival on the y-axis and follow-up in months after surgery on the x-axis. And about 44% of patients in this study expressed HSD3B1 in their cancer cells. The remainder did not. And you can see here that those who expressed HSD3B1 had much worse recurrence-free survival. Going over to this graph, same thing, Kaplan-Meier curve with disease-free survival on the y-axis, follow-up after surgery on the x-axis, and they had the exact opposite finding. In this study, they had 73% of patients expressed HSD3B1 in their cancer cells, and those people had a better survival. So none of these studies looked at androgen receptor and the interaction with that, and they also didn't look at the significance of the possible significance of the mutation. All this is to say that this is pretty much all the data that we have, and it's conflicting as to the role of this gene in breast cancer. The only studies that I've been able to find that actually specifically look at genotyping of the HSD3B1 gene and how that might affect someone who has breast cancer has to do with 2 studies looking at breast density. So increased breast density has been shown to be associated with increased risk of breast cancer. And what these studies shown, this study on the left chart here, with every acquired variant allele copy, Caucasian women actually had decreased breast density, whereas African American women had some increased breast density. This study on the right also, similarly to Caucasian women, 95% of women in this study were of European ancestry. Every additional copy that's acquired of the variant allele had decreased breast density. They didn't have any clinical significance associated with either of these studies, but basically to show that there is potentially some interaction with these allele copies and breast cancer. And that's it. That's all the data that we have for this gene in breast cancer. So my goal for research was to start out slow and essentially just establish that this gene and the variant copy actually do exist in breast cancer patients. So to establish an allele frequency, and then to move on and to look at evaluate the association between this allele and aromatase inhibitor therapy resistance. My hypothesis, much aligned with that in prostate cancer, is that women who have the homozygous variant allele, so 2 copies of the variant allele would have the worst outcomes, 2 copies of the wild type allele would have the best outcomes, and heterozygous women would have intermediate outcomes to that. So this is why I came up with this hypothesis. We again look at the postmenopausal women, and this is either women who are officially postmenopausal or premenopausal women who are functionally postmenopausal, meaning they're on ovarian suppression. They go through this extra gonadal synthesis pathway through adrenal gland synthesis of DHEA, which we talked about before. Aromatase inhibitors, again, as I mentioned previously, very important in the metabolism of these androgen precursors to estrogen. When aromatase inhibitors bind to the aromatase enzyme, you have buildup of DHT and decreasing estrogen levels. And that's essentially how these drugs work. So you have decreased estrogen receptor activation because there's no estrogen to bind to the estrogen receptor. And then it's theorized that these drugs also unmask the inhibitory effect potentially of DHT on the androgen receptor. So there is some literature that suggests that when DHT binds to the androgen receptor, it can actually inhibit breast cancer tumorigenesis. For women who have the variant allele, I would assume, like in men, that they have increased concentrations of the enzyme in the cells, which will drive biosynthesis of the DHEA through the precursors, and then based just on concentration and enzyme kinetics, meaning that these drugs are more likely to bind than an aromatase inhibitor would be to aromatase. Aromatase inhibitors will not work in these patients, and they will go through this metabolic pathway and be driven to produce more estrogen and thus bind to the estrogen receptor and have increased stimulation. So in terms of the first part of my research to just establish an allele frequency, we enrolled 30 patients in a study just to get blood samples from them to do some germline sequencing. 29 of those patients have been sequenced at this point. All of these women had metastatic postmenopausal estrogen receptor positive breast cancer. And fairly similar to what we saw in prostate, about half of women had the homozygous wild type allele, 45% were heterozygous, and only 1 patient had the homozygous variant. This gives a germline allele frequency of 26%, and this was compared to 29% in the men with prostate cancer in the multi-cohort study that I showed you earlier. And again, in the larger population, this allele frequency is anywhere from 10 to 35%. So this is good in the sense that I'm doing research on an allele that is fairly common in the breast cancer population. And then in order to actually find out the clinical significance of this variant among women and to see whether this sort of inherently makes women resistant to endocrine therapy, I'm doing 2 studies at the same time. The first on the top here is to associate, find out whether there's any associations between the variant allele and recurrence among women who had early stage postmenopausal hormone receptor breast cancer. We're going to look at a whole population of patients, some of whom were treated with endocrine therapy and some who weren't, but really significantly, we want to think about the patients who are on endocrine therapy and whether or not they were more or less likely to recur based on having the variant copy of this allele. And then the second part of the research that I am really excited about is looking at patients who could potentially receive preoperative or neoadjuvant endocrine therapy and the effect of the variant allele on the response to preoperative endocrine therapy. And I'll just talk a little bit about these. So with respect to the question about recurrence, one of my mentors is at the Hutch, Chris Lee, and he has a large study called Bravo, which is a large epidemiologic study that's a case-case study. That basically means that only women who had breast cancer were enrolled into the study. And the goal of it was to evaluate the etiologies and mutational landscape of breast cancer subtypes. And so because of that, all of these women had some sort of DNA taken, whether it was a blood sample or a saliva sample. Women who were enrolled were 20 to 69 years old, had a diagnosis of invasive breast cancer between 2004 and 2012. And there's a lot of demographic, epidemiologic, tumor treatment, and follow-up data. The primary objective of this study is to evaluate whether there are any associations between germline HSD3B1 variants and recurrence. There are 782 postmenopausal women with estrogen receptor positive breast cancer and germline DNA that we're currently working on extracting. And 67 of these women experienced a recurrence or a second breast primary event. So this study is in its nascency. We have the germline DNA, we have all the clinical data, but we have to extract the DNA and then sequence it. So we don't have tumor DNA, so we can't get the somatic information, but basically we're going to compare women who have 2 copies of the variant, 1 copy of the variant, or no copy of the variant, and see whether or not those women were more or less likely to have a recurrence. So more on that to come. And then the second kind of major part of my research that I'm really excited about is looking at preoperative endocrine therapy. So in breast cancer treatment, most of the time, most women get surgery first, and then we decide whether they need chemotherapy after that. There are patients who can get chemotherapy first, and that's called neoadjuvant chemotherapy. There are also patients who could potentially get neoadjuvant endocrine therapy, and there have been several randomized controlled trials that showed the benefit of this. Unfortunately, endocrine therapy works very slowly. So women have to be on endocrine therapy for about 6 months to really see any demonstrable effect on the size of their cancers. So 6 months is really long for a woman to wait with a breast cancer in her breast, and it's also really long for providers to wait to see if it's going to work. And so one way to get around that and to see whether or not patients with this variant will respond better or worse to preoperative endocrine therapy is something called a window of opportunity trial. This essentially means that between diagnosis and primary treatment, so in this case surgery, we give them aromatase inhibitors and then measure some biomarkers to see how well they responded. And the plan is to look at Ki-67 and see whether women who have the variant allele will have a more dramatic or less dramatic response in the Ki-67, which is a proliferative marker. Secondary objectives here are to look at whether the androgen receptor, if patients also express the androgen receptor, whether that acts as an effect modifier. We also need to look at levels of expression of 3-beta HSD1, which is the enzyme, and then quantitate intracellular estrogen and androgen levels. So this is a schematic of what the study would look like. Patient would receive their clinical diagnostic biopsy. They'd have to have an invasive ductal cancer greater than 1 centimeter, be postmenopausal and hormone receptor positive. We would obtain Ki-67 scores, ER/PR, and AR expression on the diagnostic biopsy, screen and enroll patients. We would give them aromatase inhibitor therapy for about a month prior to surgery, also get a buccal swab or a blood sample to get their germline sequencing done, and then we look at the surgical specimen, again, evaluate Ki-67, ER/PR, and AR expression, and then we also we get sequencing of the cancer itself. And so the goal here is to look at the difference in Ki-67 expression before and after receiving an anastrozole. And my assumption is that patients with the variant will be resistant to the endocrine therapy and will have a very minimal change in Ki67, whereas women who have the normal copy of the alleles will not be resistant to endocrine therapy and will have a much larger reduction in Ki67. Ki67, as I mentioned, is a proliferative marker, and it has been shown in some of these neoadjuvant endocrine therapy studies to be associated with prognosis if you have a good response in Ki67 with neoadjuvant or preoperative endocrine therapy. So because of COVID, as I mentioned, we don't usually treat with neoadjuvant endocrine therapy, even though it works, just given the time frame in which it works. With COVID, we had a sort of population that was ripe for using endocrine therapy because we were trying to avoid utilizing extra resources, taking these patients to the OR. And so in our group, we had 17 postmenopausal women who had hormone receptor, HER2 negative, invasive breast cancers, and we opted to treat them with preoperative aromatase inhibitors. Of these 17 patients, not all of them have had their surgery yet, so we received buccal swabs and have had the opportunity to sequence 3 of them, so obviously more to come, and then we have sequenced 4 of the actual cancers, obviously more to come on that as well. So of the germline sequencing, 2 patients have the homozygous wild type, 1 heterozygous, and of the 4 that we've been able to sequence the actual cancer itself, there's 1 homozygous wild type, 2 heterozygous, and 1 homozygous variant. So these patients are not the same germline and somatic sequencing. So it will be very interesting to see once we can actually get all of the patients, whether we have any signal at all as to whether the variant has anything to do with these patients responding better or worse to aromatase inhibitors during this short interval between diagnosis and surgery. So hopefully that was somewhat clear. I don't have a lot of data to show, or really any data to show, but hopefully in another 6 months, we'll have been able to sequence a lot more patients and understand the significance of this gene in breast cancer patients. I'd like to acknowledge my mentors, VK Gotti, who has subsequently left me for Chicago, but remains actively involved in mentoring me and also in this research. Chris Lee is my mentor at the Hutch, who has so kindly allowed me to use his DNA samples from his larger epidemiologic studies. Kalyan Bond is a medical oncologist here at UW who's been actively engaged in this research, and Tina Lockwood, who's in lab medicine, who's been helping with the sequencing. Also, I have grant support through Swim Across America and the Institute of Translational Health Sciences, and so it's been very helpful to have some independent funding. Also, I should have put Department of Surgery, as I have funding through the Department of Surgery in terms of startup funds to help with some of this translational research work. And that is all I have.
Fantastic. Thank you so much for sharing with us this morning. We are right about on schedule, and so I'm going to open up the floor for anyone who would like to do actual questions. Otherwise, I will moderate chat for questions after that. Dr. Byrd.
Good morning, Megan. Fantastic talk. And my chat function, by the way, is not working. I can only privately chat to one individual on the list, and that's why I'm not using chat. I know Ben Anderson and I were smiling as you were giving your presentation because Roger Moe, who many people on this call know quite well, was our mentor, and he for decades was shouting out the androgen receptor in breast cancer is something you've got to look at, and deaf ears everywhere. If he was listening to you, and I'm sure he is, he would be grinning so far his mouth would break apart. Fantastic talk. I know the frustration you've gone to just to try to get your research going. COVID happens, VK Gotti leaves. We are with you. We support you. I love the work. Thank you so much for doing this.
Thanks. Yeah, I think the androgen receptor is something that's very understudied in breast cancer. So this research, I think hopefully if we can find some sort of significant difference in all these various, with the variants, looking subsequently at more specifically at the androgen receptor will be very interesting. I had more slides to share, but didn't because I wasn't sure where I was on time. But also triple negative breast cancers, there's a subtype of triple negative breast cancers that are androgen receptor positive. And when you look at their molecular profiling, they actually look potentially more like hormone receptor positive breast cancers than triple negative breast cancers. And so it'll be really interesting to look at whether and how this gene mutation acts in those patients, specifically because those patients also can be treated by the drugs that I mentioned for prostate cancer patients, the 17 beta 1 inhibitors and the androgen receptor inhibitors. And in metastatic patients with these AR positive triple negative breast cancer patients, we know that there's actually a survival advantage associated with androgen receptor blocking. So, yeah.
Dr. Anderson?
I really agree with David. Wonderful talk, Megan, and very logical. I like the way that you're walking us through. We could follow the rationale and the going to clinical therapy. I have two questions. The first is that the mutation, the heterozygotes, the homozygote wild type versus variant, we're seeing a difference in responses to therapy. Am I gathering correctly that this does not relate to tumorigenesis? Doesn't relate to people being predisposed to cancers, whether it be prostate or breast. It's really, this is just a response area. Is that right? It's not, well, this is not a gene mutation that we should be thinking about prevention or that sort of thing. Is that right?
No, I mean, not in the same sense, I think, as BRCA at this point, but one could postulate that, looking at the prostate data, if someone is born with a germline, 2 copies mutated and they have the CC variant, they have all of this extragonadal synthesis of DHT, and DHT is an androgen receptor agonist very potently. And so the research really hasn't been done because Nima Sharifi, who first established that this SNP existed in prostate cancer patients, this was discovered in like 2013, and they just did all of the clinical trial, studies in 2016, 2017. So I don't think they've had enough data to yet look at whether or not this actually acts in terms of promoting the prostate cancer in the first place. But I guess I wouldn't be surprised if it did. And this is now standard sequencing in prostate cancer. So when they do Oncoplex, which is a test that we have here at UW that actually does somatic sequencing of all of the tumors in prostate cancer, HSD3B1 has been now added to Oncoplex because it's so clinically significant.
So you mentioned the breast density data, and breast density has to do with incidence of cancer as opposed to therapy. My other question or point, you're trying to harvest data from available sources. That's, going to Chris Lee, I think it's brilliant, going to Chris Lee's Bravo study. The other area, you're looking for Ki-67, which now we're not testing as an isolated measure anymore, but we are measuring Oncotype DX often, and Ki-67 is a prominent gene within Oncotype. And another low-hanging fruit opportunity might be to see, and it's a little bit of a Hail Mary pass, but is there any correlation between gene expression and Oncotype? Because that might really give you an avenue into this that would be therapeutically relevant. You mentioned using chemotherapy. Should I use chemo? Should I not use chemo? And so that it might be an area that you could go to and to look for funding if there was some preliminary data that you could get, because Oncotype is in the chart.
Yeah,
that's great talk.
That's a really good point. I actually hadn't even thought about that. Thank you.
Sorry, one more point. I'm sorry for dominating this. You talked about how nobody's doing endocrine therapy for 6 months. Kelly Hunt from MD Anderson gave a really good talk, virtual talk at SSO about the endocrine therapy and thinking about 6 months, because it does take a long time. I think we're going to see more of this, particularly with this COVID thing where we've been doing this, and you may have opportunities in trials. And talking to Kelly Hunt might not be a bad idea, given that they've got gazillions of samples for everything. Anyway, thanks so much. Sorry for dominating.
No, thanks. That's a really good point as well. It's neoadjuvant endocrine therapy is frequently used in Europe. I don't know that maybe they're more patient than we are here. But I do agree. I think that, just with COVID, we've seen that it is safe. you know, really to use. And it may be that we start using it more often, because it would be nice in some of these, as you and I and all people who do breast cancer surgery, if people have positive lymph nodes and they're ER positive, we don't tend to give them neoadjuvant chemotherapy. We just dissect their axilla. If we could downstage their axilla with neoadjuvant endocrine therapy, we could potentially avoid some of the morbidity associated with maxillary dissection. So it would be nice to be able to get this more into the mainstream. And if we can, you know, sort of preoperatively identify patients that are more or less likely to respond to neoadjuvant endocrine therapy, you know, it would be great to be able to stratify those patients up front.
We definitely are open for more verbal questions, and the chat box, I apologize, it was not open. It is now open, should someone want to use it.
Meghan, it's Sarah. I want to congratulate you on really a huge amount of work that you put into this and developing an idea, a research idea, and generating a hypothesis. Given the papers you presented, you're on the cutting edge and the forefront of this area, domain in breast cancer research. And I'm glad you have the support of some of our medical oncology colleagues in moving this forward, as well as at the Hutch. I had a question regarding some of the background research you presented that shows a kind of polar opposite results, as you said, with disease-free progression, depending upon the 3-SHB positive, negative. And so can you kind of postulate why that might have been shown to be the case, that those two study results were so different?
Yeah, so those two studies, you know, I presented them just because I had to present what's out there in the literature. They looked at all different kinds of subtypes. So they looked at hormone receptor positive and negative, triple negative, HER2 positive, right? And so even just looking at the expression of the gene in the cancer cells, one study, 44% of the 44% of the patients expressed HSD3B1, and the other one 70% did, right? So the populations are clearly not really comparable. And they also didn't assess androgen receptor positivity at all, which I suspect has some significance in this whole pathway. So I don't really know what to make of it, except to say that they're probably just potentially different patient populations, and that we kind of need to know more, I think was the take-home point there.
Makes sense.
I see Teresa's question, if hormone therapy induces an immune response or change in intratumoral immune infiltrate. I assume that it probably does, but I don't know exactly what. So definitely something that if we're able to show that there is a clinical significance, you know, looking at sort of intracellular levels of different hormones, and then also looking like what you're asking about immune response would be really important. We know.
Yeah, it's so great that you're doing this. It's amazing that you've gotten a trial up and going in the short bit of time that you've been here, and you'll have access to tissue to do all kinds of studies, which is great.
Yeah, maybe. We'll see.
We believe in it.
Yeah. Well, thank you, everyone.
Yeah, thank you, everyone, for being here. So thank you, Dr. Flanagan. Fantastic presentation. We'll look forward to seeing everyone shortly. Harkins, I think, is our next large meeting, and then Grand Rounds next month. Have a great week, everybody.
Thanks.